Good afternoon, everyone, and welcome to today's webinar. Next slide, please. My name is Tapushi Patel, and I am the Frontline Territory Manager from Nutricia for the state of New Jersey. I am a registered dietitian specialized in pediatrics, and I'm equally excited to be here today with you all and learn from our expert speaker about practical management of infants with cow milk allergy and the role of gut microbiota. Are we doing enough? Next slide, please. We're lucky to have with us Dr. Kelly Tappenden. Dr. Kelly Tappenden is professor and head of Department of Kinesiology and Nutrition at the University of Illinois at Chicago. She has received multiple awards, published over 100 peer-reviewed papers, and delivered over 400 invited lectures. Dr. Tappenden has lectured across the U.S., sharing her expertise and unique insights related to the role of gut microbiota, pre- and probiotics in the management of pediatric allergy. She has served as the 33rd President of the American Society for Parenteral and Enteral Nutrition in 2008 to 2009, as a chair of the Nutrition, Metabolism, and Obesity section of the American Gastroenterology Association Institute from 2009 to 2013, and presently represents the American Society for Nutrition on the Federation of American Societies for Experimental Biology. Dr. Tappenden has been the editor-in-chief of the Journal of Parenteral and Enteral Nutrition since 2010. We welcome you, Dr. Tappenden. The stage is all yours.
Topic today. I also want to thank Nutricia for sponsoring us today. They did provide me with an honorarium, though they did not dictate this presentation. I made this on my own. So what I really want to do is spend some time talking about the gut microbiota. We're going to focus on early life and the development of the immune system, but it applies to the whole life. The microbiota has similar physiological functions throughout life. We'll then talk about specific probiotics and prebiotics with an emphasis on prebiotics, because when we talk to our clinician colleagues, they all talk about probiotics, right? But prebiotics is a little bit more of a new area, or overlooked anyways for many of them. And then I'm hoping we can talk about practical tips that would be supported by the evidence that any of you may be interested in. So I want to start off with a poll. There's going to be two questions that are going to pop up. First, are you currently using pre or probiotics in your clinical practice? Yes, prebiotics. Yes, probiotics. C would be yes, pre and pro, what we know as Synbiotics, right? Or no. The second poll that I want you to answer is: the gut microbiota in infancy supports digestion and impacts maturation of the immune system. So how strongly do you agree or disagree with that statement? We'll give it a moment and then we'll be able to see the results. Okay, the first one, how much you use pre and or probiotics clinically, and then the second, the gut microbiota, so it's digestion and maturation of the immune system. Okay, so here we go. This answer to the first question is really interesting to me, because what you can see here is that we have a number of individuals who don't use it, about a third, and then about a third again use both. So there are very few people who exclusively use prebiotics or probiotics, but either they don't use them at all or they are using both. And many of you strongly agree that the intestinal microbiota supports digestion as well as development of the immune system. And I'm quite glad for that. That's certainly what we're going to be talking about today. Let me remind you that the human body, and the intestine in particular, is home to trillions of microbes, mostly bacteria, but also yeast, protozoa, fungi. they outnumber cells in the human body by 10-fold. In fact, it weighs up to 2 kilos or close to 5 pounds. And they interact on this vast surface area. You will have heard all sorts of ways that the surface area of the intestine is described like a tennis court. But it's also 20 times that the surface area of our skin. So that might be a new one you haven't heard of before. That single layer of epithelium lining our intestine and separating us from the lumen, which is the external environment, also contains 70 to 80% of our immune system. And some people wonder about, why do we hear microbiota, microbiome, microflora? Micro, of course, means small. Flora refers to plants. And so that you won't see it much in the literature anymore because that's not correct. Biota refers then microbiota to small life. Now, the difference between microbiota and microbiome is that biota refers to the population of bacteria that are present. Microbiome refers to all the DNA of those microbes that are present. And so microbiome would be more accurate if you were talking using various molecular techniques to describe what was present there. From a DNA perspective, microbiome would, or microbiota would be more accurate if you're talking about the life that exists there. At any rate, we know that these microbes are critically important, important for many aspects of human health. They do help digest certain foods. Dietary fiber would be a great example of that, and they produce nutrients such as short-chain fatty acids or butyrate that are really very healthy. Butyrate's primary fuel for our colonocytes, number one preferred fuel. They also produce vitamins, B12, folate, vitamin K. This was discovered early on in nude-free animals, where animals were born and reared in a sterile environment. And in order for those animals to grow at the same rate as the control or conventional animals that did have normal development of the microbiota, in order to grow the same rate, they needed up to 30% more energy and supplementation of various vitamins to be able to meet their needs. We also know, though, that the microbiota is really important in regulating energy metabolism, right? There's more and more information now regarding the impact of the microbiota on obesity, the gut-brain barrier, so it has psychiatric, central nervous system implications, and then immune, the second part of that question, which many of you strongly agree with. The nude mice, those germ-free mice, they develop infections, or they're susceptible to intestinal pathogens much more than their control counterparts. So the microbiota has been shown to defend against harmful microorganisms, as well as also be able to develop the immune system. So let's talk about that a little bit more closely. What you can see here is there's two concepts. So the microbiota themselves are acting in a way to protect the host, and that's called colonization resistance. You can see here in the intestinal lumen, there's a mucus layer that lies over that single layer of epithelial cells that produce the mucosal surface area in our intestine. And having a healthy, balanced microbiota there is really important for this concept of colonization resistance. And why that will prevent the development or the susceptibility to intestinal pathogens. The healthy bacteria are competing for the same resources as the pathogens are, whether that be fuel within the lumen, epithelial receptors on the epithelial cells. The epithelial themselves cells are secreting antibacterial substances. So the presence of that microbiota in a healthy and a robust way is one of the things that does help prevent against pathogenic infections. You know, the best way to remember colonization resistance, in my opinion, is C. diff disease, right? When are patients or any of us susceptible to C. diff infection? It's right after we have antibiotics, right? The microbiota has been reduced. The diversity has been reduced due to the antibiotics, and that then gives the opportunity for pathogens like Clostridium difficile to infect, because there's room and they have all the resources on their own. So there's another aspect, though. It's not just the microbes that are doing this work. The microbes are actually helping our intestine do the work too. Okay, so let me remind you that 70% of all immune cells are in our gut, in the gut-associated lymphoid tissue. So we'll have intraepithelial lymphocytes that will come and wedge their way in between enterocytes, panocytes, the endocrine cells of the gut. And they are sampling then what's happening in the lumen. They're interacting with that normal healthy microbiota, and that helps develop the immune system in a way that enhances host defense. It also, though, it doesn't just develop the immune system and make it ward off things better. It makes it able to discriminate. And so oral tolerance is highly dependent upon a development of a healthy microbiota too. You can see here in this image, we have a bar graph showing dark blue Bifidobacteria bacteroides. The light blue is firmicutes, and then the purplish color is Proteobacteria, Enterobacteria. The left two bars are microbiota sampled in infants at 3 months. The left, those who do not develop allergies, and those who do. So that atopic march where we're looking at the atopy that may come on the skin, we know that then leads to allergies in many cases and continues on to asthma. That process is associated with fewer bifidobacteria when looked at when the microbiota is sampled at 3 months. If we look at 1 year, there's still changes in that microbiota. And so what's being shown over a number of studies is that reduced richness, diversity at 3 months of age is associated with those diagnoses, with atopy and cow's milk allergy in this case at 1 year of age. So it's interesting. The changes in the microbiota, the negative changes, what we call dysbiosis, are associated with these diagnoses later on. And this is not just the only study. You can see here, I've shown several studies here on this slide where you can see that the phenotype or diagnosis is well after the age at which the dysbiosis is diagnosed. Okay, so in children 1 month of age that will go on, they'll have a dysbiosis, and they will have a higher rate of eczema at 2 years. Dysbiosis at 1 week is predictive of eczema at 18 months. You can see many of these different phenotypes of the atopic march associated with this. Here at the first week of life, in the first week of life, a dysbiosis there is positively associated with you can see IgE, EOS, rhinitis, eczema at 6 years of life. Okay, so it well precedes it. Now, I don't want anyone to leave this saying that dysbiosis causes those things. What we have here is an association, but we have an association that's been really well measured in many studies and thousands of infants at this point. So if that's the case, if a dysbiosis early in life, even in the first few days of life, is associated with these diagnoses later on, it's attractive to think if we can prevent that dysbiosis early, can we reduce the risk of that diagnosis later on? And that's a hypothesis, okay? It's a hypothesis that's supported by the literature. What you can see here is that early life really is a window of opportunity for us to help that colonization process go well. During pregnancy, we know that there is initial seeding of the microbiota through the mother's placenta and amniotic fluid. that was something that we always thought to be sterile, right? It's now known that the placenta and the amniotic fluid have a microbiota. We know that at birth, there's a very big exposure to microbiota, whether it be because of mode of delivery or gestational age, and then depending on the circumstances of that child during early infancy and continuing on till about 3 years of age. There's many factors that can predict the development of a healthy versus dysbiotic microbiota. So you will know that the mode of birth is important, whether or not a child is vaginally versus cesarean derived, whether or not that child is going to be fed a certain way while they're in their first year of life, whether they're getting breast milk or formula, introduction of solid food, and during the toddler years, 1 to 3 years, as they start to consume a full diet. There are many maternal factors, all right? So what the mom's microbiota is like, whether or not there's a normal vaginal microbiota, mode of birth as we discussed, and then there's postnatal factors. So this would include the antibiotics, breastfeeding, host genetics, the environment. All of these things have an impact and play into the microbiota that develops. This is a paper that I wanted to reference for you in Nature Medicine 2016 that goes through a number of factors that are viewed to be disruptive for the microbiota. So you can see here, large studies, a 1.9 million child study in Danish children from birth through 15 years of age, okay, looking at an outcome that's associated then with various factors that are known to disrupt the microbiota early in life. So cesarean section, antibiotic treatment, probiotics, diet supplements. The diet supplement in this case would be iron supplementation, in hygiene, pets. All of these issues are known to impact the microbiota development and thereby impacts then the physiological protection that's provided by that hopefully optimal microbiota or certainly less than optimal in many of these cases, right? Let's talk about diet. We know, of course, for many reasons, that human milk is best for infant health. There are many, many different factors associated with better outcomes when a child is receiving human milk compared to formula, right? I don't think we need to review that. But the microbiota is one of those factors too. Now, if we look at human milk, what is it that's so beneficial? And we're going to focus on the microbiota because we could have a lecture just on any one of those factors, right? The composition of human milk appears to be really important, and it's quite complex. So HMO stands for human milk oligosaccharides. That's really, really important. You know, the other thing that we've found is not sterile is breast milk. Breast milk contains a microbiota that is seeded actually from the mother's intestine. During the insult of labor, there's thought to be dilation of the tight junctions when there's a transfer then of some of the microbiota from the gut to the mammary gland, and there is live bacteria, a microbiota in human milk. There's special fats, as you know. Lactose is the primary sugar. We have protein, hormones, vitamins, living cells, nucleotides, minerals. It's a very complex fluid that's provided to fuel human infants, right? Let's think about the bacterial composition then. I don't know if you were aware that the microbiota contained, or the human milk contained a microbiota. That's something that's actually relatively new. You can see that there were some early papers in the first decade, or from 2003, 2009, 2011. Many clinicians that I talked to, even in neonatology, are under the impression that infants are born sterile and that human milk is sterile. It's not the case. What is the case, though, is that there's a lot of variability between the composition of microbes in human milk. And that's because each mom is varying. All of the circumstances and the impact on her has been varied. So it varies a great deal between mothers, and it also varies over time, much like we know the lipid content varies from colostrum through to mature milk. So does the microbiota content and composition. But you can see here that human milk contains on order of 10 to the 3 to 10 to the 5 CFUs per mil. And it contains lactobacilli, bifidobacterium, staphylococcus. If we look at various genus, there will be species in there that are going to be present and viewed to be beneficial in human milk. There's also then a prebiotic. So if we think of the microbes as a probiotic in human milk, the live bacteria that's being fed to the child, the HMOs or human milk oligosaccharides can be thought of as a prebiotic because what they do is help fuel the microbiota's development. So HMOs are carbohydrates. There are thousands of structures, and we are early on in the identification process. Okay, we've identified less than 200. Some are short chain, some are long chain. They're all being characterized. They vary amongst moms. Some are sialated, some are not. What you can see, though, is that they're present at a level of 12 to 15 grams per liter. Okay, we talked about protein and why we always make sure that children get their protein. If you look in human milk, there's actually more prebiotic HMOs on a concentration basis than there is protein. That speaks to how important HMOs are. Lactose, you'll know, will be the major sugar or carbohydrate, but prebiotics are also very, very important. And they're found to then fuel the development of the microbiota. They are the substrate that the microbiota ferments, produce various factors that then the next set of microbiota will ferment and helps then provide resources for that community. They allow for the growth of the beneficial bacteria, but they're also important for brain development, direct effects on the immune cells, and anti-infective effects as receptor analogs. This one is really interesting. There's HMOs that have been shown to reduce the infectious rate by a considerable amount of things like norovirus, because they're able to bind to act as decoys, the receptors on the various pathogen, and then will prevent the infection from occurring. So really, really interesting work there. And it explains why HMOs in the literature are associated with reduced pathogenic infection and reduced diarrheal diseases. So human milk is so important. And I think we can look at that live bacteria that's produced and the prebiotics that's provided too, and think of it as the ultimate symbiotic for human infants. Now, the issue is that many infants will not receive human milk, right? And so we give them formula for many good reasons, and there are very good formulas where attempts have been made to make sure that they are producing the same effects as human milk as much as possible. There are differences, though, in the microbiota between children that are receiving human milk versus those that are receiving formula. And the biggest difference is that a child fed human milk has high levels of bifidobacteria. You can see the legend here on the right side. Very high, stable levels of bifidobacteria during that period of infancy. As one develops through age, they'll become more diverse. And an adult, of course, would want a very abundant, diverse, stable community. In infants, that stable bifidobacteria population appears to be associated with the most benefit. and that's achieved with breastfeeding. Now, formula feeding produces or is associated with a microbiota that's far more diverse, much more adult-like, changes much more based on the environment. And so it doesn't give that important bifidobacteria to the same way. It doesn't foster that, nor does it give that stability. And we know that children-fed formula have higher levels of infections, right? So it's very attractive then for us to hypothesize and seek to use a symbiotic approach with formulas wherein we add the appropriate prebiotic, we add the appropriate probiotic to help foster the intestinal microbiota and optimal development of that, because that might improve clinical outcomes in children, right? And there's some work that's been done looking at that. Prebiotics have been added to infant formulas and compared to standard formulas and then human infants. When it comes to infant formulas, the most common prebiotics that you'll see used are short-chain galacto-oligosaccharide, long-chain fructo-oligosaccharide, often called inulin. That doesn't mean these are the ideal options or the only options, but this is where most of the research has been done. There are hundreds of structures, though, that could be used as prebiotics in formula. So when we look at that literature, there are many trials. This is a review that looks at 6 clinical trials, a systematic review looking at galacto-oligosaccharide and fructo-oligosaccharide, short and long chain respectively, supplementing in infant formula. Okay, so when a prebiotic supplemented infant formula is compared then to control, which is breast milk in this instance, you can see that breastfed infants on this image have nice amounts of bifidobacteria, and that's going to be more similar to breastfed infants. Then the same can be said for lactobacilli. Increasing the levels compared to the control formula of both bifidobacteria and lactobacilli, and importantly, the breastfed reference group, which is identified by this square, is showing that by supplementing the infant formula, you're achieving the same level of these beneficial microbes. So very important data. It also discourages the growth of potential pathogens. This was a study done in preterm infants, supplemented with the same prebiotic, a formula supplemented there. It decreased the sum of the pathogens. So in this instance, it was cultured, the amount of pathogens were cultured, and you can see when the prebiotic formula was fed to a group, they had fewer pathogens in their microbiota compared to infants consuming a cow's milk formula. Okay, so that's important for sure. Does it impact then the infections that those children are seeing? And the answer is yes. Here you can see a study done in healthy infants with infant formula, a premium infant formula as a control versus that same infant formula supplemented with prebiotics. And what you can see here on the right is that the prebiotic formula resulted in lower incidence of infections over time during the first 2 months of life, during the second to fourth month of life, and during 4th to 6th months of life. The blue bar representing the incidence of infections in these children or infants is reduced compared to the infants consuming the control formula. So the prebiotic supplemented formula was reducing that incidence of infection, a very important clinical outcome, right? Now, the interesting thing about this study is these authors went on, and after the 6 months this study was done, the children went to consume whatever it was that was offered to them, and they chose to. But the authors still followed these children. They followed 134 of them and looked at whether or not prebiotics in formula during that first 6 months of life would have a lasting effect over the first 2 years of life. So long after that formula had been stopped, was there still a benefit? And you can see here that yes, the prebiotic supplemented formula resulted in a group with lower numbers of overall infections here in the first two bars. Blue, of course, being the prebiotic containing formula compared to the control formula. Upper respiratory tract infections, as well as any infection requiring antibiotic therapy. There were reduced infections of any kind, but specifically upper respiratory tract infections were markedly and statistically reduced. A nice benefit, right, for having that prebiotic during the first 6 months of life. It wasn't just infections, though. They also looked at allergic manifestations and found that there was increased incidence in the control group. So the prebiotic formula was protective with atopic dermatitis, wheezing, and hives. So we were talking about the atopic march. Children who consumed a prebiotic containing formula during the first 6 months of life had reduced incidence of those diagnoses by age 2, pardon me. When we look at intestinal infections, this is a study that did so looking again at a prebiotic supplemented formula compared to a controlled formula in healthy infants, and that they found that the number of episodes of gastroenteritis was reduced. You can see here by 50% or more, and that when a diarrhea did develop, it was viewed to be 58% less acute. Okay, so the duration of if diarrhea did develop, which happened less often in the children consuming the prebiotic group, if it did develop, the duration of that was reduced too in this study. So beneficial effects on the intestine. There's also been some work done in the Netherlands looking at then healthcare economics and whether or not there's a real benefit to having these prebiotics in the formula. What this group did is look at the various diagnoses associated with atopic dermatitis. So given that atopic dermatitis, we know then marches on to these other diagnoses. If groups were to be consuming prebiotics versus not, this group in the Netherlands constructed a model looking at published literature, clinical trials, official price and tariff lists, national population statistics. And what they were able to find is that prebiotics cost 51 euros at the time the study was published, which was 2012. There was a 0.108 adjusted life years. So that means that the quality of life while the child was consuming this formula was improved. This amount, 0.108 years, so just over a month, right, was improved, and that there was an incremental cost effectiveness ratio of 472 euros per child. So we're talking here about a relatively low cost prebiotic formula being able to improve not only the diagnosis of these things as we looked at in the past studies, but improve the quality of life that is perceived and assessed from the parents, and that there's also a big cost savings too, even when you take into account that the prebiotic formula may be more expensive or incrementally more expensive. So when this comes to children then with allergies, we know that we want to make sure that we are using a formula that would eliminate allergens, right? So a hypoallergenic formula, 100% amino acids, so that there isn't development or trigger of that allergy by allergens during the first 12 months of life is going to be standard therapy. However, in those children where we're wanting to use this formula, we can't just be throwing out all the benefits that we know are associated with human milk that has live bacteria as well as then prebiotics, as we've talked about, because in that instance, particularly children at high risk or already having some diagnosis involved with atopy, that we want to be ensuring that we support development of the gut as much as possible, right? So that leaves us then to look at these data and see that a prebiotic supplemented formula, there's going to be strong rationale for that. And also containing a live bacteria, Bifidobacterium breve. This is a live bacteria, so acting as a probiotic in the formula. And of note, this is the most common Bifidobacterium species that is present in both human milk as well as the human intestine. So very relevant and also safe bacteria. What happens when a formula like this is fed to children who require an amino acid-based formula due to allergic manifestations or high risk thereof? So what you can see here is a study that was actually conducted by our sponsor today, Nutricia. But listen, don't just regard this study because of the funding source. What Nutricia did was look at the Synbiotic formula, a Synbiotic Neocate, compared to Neocate itself. So their control was their own formula. This was not to say our formula is better than someone else's formula, but to look at their own amino acid-based formula and say, can we do better than that if we add Synbiotics? And so you can see here the test product, the Neocate with Prebiotics. I believe it's called Neocate Cineo, though we'll have to ask Takushi to be certain, versus the control Neocate for 8 weeks. And then there were samples that were done at follow-up periods. On the left, you can see Neocate compared to Neocate Cineo. Yes, it does say that on this slide. At week 0, there was no difference. However, at week 8, so after consumption of the formulas for 8 weeks, you can see that there was increased abundance of Bifidobacterium species. And what this gray shading is behind is what would be present in children consuming human milk. So to say that again, if we looked at the feces, the children or infants consuming the control Neocate had less abundant Bifidobacteria than those consuming the prebiotic supplemented Neocate, as well as the breastfed infants. And the prebiotic supplemented Neocate increased that Bifido to the level of those children receiving human milk. So a very important result. You can also see the opposite happens, which is also beneficial when you look at pathogens. Okay, no difference at baseline, but after the 8 weeks of treatment versus control, you can see there were fewer pathogens shown or negative bacteria shown in the feces of the children consuming the prebiotic formula compared to standard Neocate, and it was reduced to the level of humans consuming breast milk, what we view as the gold standard. So really, really important data. There's a recent meta-analysis that looks at all of the papers related to amino acid formulas and a Synbiotic supplemented formula. I have these here for your interest, but let me just get down to Sorensen. If you're interested in looking up all these data, Sorensen is the first author. It was published in Nutrients just a few weeks ago in 2021. Okay, so you'll be able to look at those papers and see the benefit over and over again. What that meta-analysis does, if I just cut to the results here, is show that this amino acid formula supplemented with Synbiotics had an increased percentage of fecal bifidobacterial species and then reduced percentage of adult-like species that we don't want to be seeing in these children. So like the study I showed you, there are many other studies in the literature that show similar results, okay, by using an amino acid-based formula supplemented with pre and probiotics. So I want to conclude here by just saying that nutritional strategies employing both probiotics and prebiotic fibers, so Synbiotics, as about a third of you are using, is really important for addressing dysbiosis in the developing intestinal microbiota in human infants, and stimulate critical development of the immune system that's associated with then enhanced outcomes for those children, whether it be reduced infections, reduced allergic manifestations. There's benefits here. So I want to leave some time for Tapushi and hope that we are able to discuss this a bit. But please, when you are thinking of the children that you are feeding and prescribing formulas for them, remember the microbiota. Remember how important it is to the health of the child, not just at that time, but years down the road, and see what you can do to ensure the presence of prebiotics and probiotics. Tapushi, I'll give you the floor now.
Hi, Dr. Tappenden, it's Rachel here. I'm going to...
Oh, pardon me, Rachel.
Yeah. So thank you very much for that presentation. That was really informative. And just to our audience, if you have any questions for Dr. Tappenden, now's the time to start typing them into the Q&A box down in the bottom right-hand side of the screen. And just while you're doing that, Dr. Tappenden, if you could forward on to the next slide.
Absolutely.
Thank you. Okay, the one before that. Sorry, if you go back.
The slide before that with the, yes, okay, yes, that's where we were.
Okay, yes. So just a couple of housekeeping remarks. We would love to hear your feedback on this webinar, and as a result, we have a very short survey that you can access through the link on the screen or by holding up your camera to the QR code on the screen, and that will take you straight through. And if you are looking for a CE certificate, there'll be instructions at the end of the survey for how to receive that. And so thank you in advance for your feedback. Okay, so I'm just going to look to see if there are any questions. Okay. So while you're thinking of those questions, I might just kick off with some questions.
Sure.
The first one I have here, Dr. Tappenden, is, so when you're thinking about practical tips for RDs, what advice would you give to registered dietitians, when they're trying to choose a formula with probiotics or prebiotics for their allergic infants? What would you look for?
Sure. So I think what you're going to want to be looking for is the type of pre or probiotic and the abundance that's there. When it comes to probiotic, of course, the data I just showed you was Bifidobacterium breve, and that is a very reasonable choice, particularly the species used, because it's so abundant in human milk as well as infants' microbiota. When it comes to prebiotics, you want to be thinking about diversity, and when it comes to prebiotics, length often makes a difference. So there's various kinds of prebiotics. We were talking about galacto-oligosaccharides and fructo-oligosaccharides. There's also some that are dietary fiber. The rate of fermentation appears to be really important. So a short chain prebiotic will be fermented much more rapidly than a long chain prebiotic will on a weight per weight basis, because what happens is the microbes will start fermenting on the end of that chain, and if you have 10 grams of a short and a long chain, you will know that there will be many more particles within the short chain to make up that 10 grams. So bacteria and microbes can start fermenting very quickly on that. They ferment quickly and will be providing short chain fatty acids and an ideal population in the proximal portion of the colon and the ileum. Don't remember, it's not just the colon. A long chain prebiotic is going to be fermented much more slowly along the length of the gut and provide then this fuel for the microbes, but then also the substrate and metabolic products that are beneficial for the other microbes as well as us as a host along the length of the gut.
Okay.
Oh, let me just say one more thing. Usually you want to target between, most of the data have looked between 4 and 8 grams per liter. And that's going to be important. You don't want to go too high. If you get up in the 20 area, you'll induce osmotic diarrhea. Because remember, these particles are not absorbed. They're fermented. They will stay in the intestinal lumen. So if we put too many in there, that can cause some osmotic secretion from the epithelial cells and lead to diarrhea. So the concentration is really important also.
Okay, great, thanks. Now this is a question from Ellen. She is asking you, what did you say HMOS stood for in your presentation?
Sure. Human milk oligosaccharides. So when we think of carbohydrate being polysaccharides, long chains, or sugars, of course, small chains, the medium chain is an oligosaccharide. Just like a medium chain peptide would be an oligopeptide link, right? So human milk, being that they're found in human milk, oligosaccharides is what HMO stands for.
Okay.
Thanks for clarifying.
All right, so another question for you. Do you suggest continuing this in older children and adolescents?
Oh, I'm so glad you asked that because I think it's so important. And, you know, I think what we have to do is look at the DRIs, you know, groups of very well thought individuals, very learned individuals have determined what human needs are, right? And we can, for nutrients, we can debate the exact level, but since 2020 or 2000, pardon me, for 20 years now, we have had a DRI, a dietary reference intake that includes an RDA and everything we as dietitians know for dietary fiber. And just like we do for dietary protein, for essential fatty acids, there is a recommendation that individuals, children through the elderly, should be consuming and is associated with better health. So when we're picking a formula for an individual, a patient, I really think we need to be at the point where fiber is included as a prebiotic or as a dietary fiber, where there's some kind of prebiotic blend as the standard and not as an exception that we're only doing if we don't, you know, we're trying to find something to work. That should be our first line formula, in my opinion. And that's supported by the fact that each of us should be consuming 14 grams per 1,000 kcals that we eat each day, right? Dietary fiber is essential. We need to stop choosing formulas as our first round that don't contain it.
Okay, well, thank you for that. Okay, so I have another question here. So this is a question from Jennifer. I've read recently that probiotic supplements are pointless because they're not able to colonize the amount of bacteria already in the gut. Can you comment on this?
Yeah. Did you say Jennifer? Was it Jennifer? Because I love that question.
Jennifer, yes.
They are not pointless if you look at specific probiotics and specific diagnoses. But just throwing a probiotic at someone for, you know, haphazardly, whatever they pick up at Walgreens for whatever purpose they may be thinking it'll work for is not going to be useful at all. And the other thing is, even if we are using a probiotic where there's evidence, so let's talk about Bifidobacteria infantis in irritable bowel syndrome. We have good data to support that practice. The benefit will be seen while the individual is consuming the probiotic. So we need to understand that when we consume probiotics, they do get into our microbiota and they have a beneficial effect while they're interacting with the microbes there, but they're passing on through. They do not, in really the vast majority of the literature has shown that they do not take up resident and become a permanent member of the microbiota. So Jennifer, you're absolutely right in that there's no lasting effect. Compare this to the data we just looked at for prebiotics, wherein children consuming a prebiotic formula in the first 6 months of life have used that to establish a microbiota that's giving them protection 2 years later. I think that that's really one of the things that's so powerful about prebiotics, and us as nutritionists need to be using. There's beneficial effects of prebiotics months and years down the line, and that hasn't been shown for probiotics.
Okay, thank you. So looking at the time, we might wrap up the Q&A session. So thank you very much for your presentation and for answering the questions. And thank you to the audience for attending and for asking your questions. This now concludes the CE eligible portion